So welcome to today's RNA Collaborative Seminar hosted by the National Cancer Institute RNA Biology Initiative at the National Institutes of Health. I'm Sandra Wolin, and I head the RNA Biology Initiative. Today you will hear talks from two of our investigators. There will be questions and answers at the end of each talk, so please write your questions in the chat. Our first speaker is Dr. Shua Gu, an investigator in the RNA Biology Laboratory at the National Cancer Institute, National Institutes of Health in Frederick, Maryland. Shua was an undergraduate at Tsinghua University in Beijing, a graduate student at the City of Hope Medical Center, and a postdoctoral fellow at Stanford University with Mark Kaye. He arrived at the National Cancer Institute at the end of 2013.
All right, so thanks for the introduction, and thank you for giving us the opportunity to present our work here. My group studies microRNA biogenesis and function. Today, I will talk about how 3' uridylation modifies microRNA function. So this slide shows you microRNA pathway. Instead of giving a detailed introduction, I just want to emphasize that microRNA need to bind to organelle protein to form the RNA-induced silencing complex, or RISC, to perform their function. The association between RISC and the target mRNA is necessary and often sufficient to trigger the inhibition of target expression. Therefore, it is critical to understand how microRNA recognize the target sequence. The region counting from position 2 to 8 from the 5' end of a microRNA is called the seed. Pairing this region to the target is essential for microRNA function. In contrast, pairing of the 3' region is often dispensable. The importance of the seed sequence had several implications. First, the function of a microRNA is mainly defined by the seed sequence. Any change to the pairing of the seed usually reduce or eliminates microRNA function. Second, since the seed sequence is only 7 nucleotide long, each microRNA can regulate hundreds of targets. In fact, more than 60% of the protein coding genes and nearly all biological pathways are likely under microRNA regulation. Not surprisingly, dysregulation of microRNA is associated with many diseases, including cancer. It was originally thought each pre-microRNA can encode up to 2 mature microRNA, one from the 5' arm, one from the 3' arm. Now, with deep sequencing, we know each microRNA have many isoforms which differ in length and sequences. Here is one example. This is the canonical sequence. When isomer gain or loss nucleotides at the 5' end, they are called 5' isomers. Likewise, 3' isomers are those buried at the 3' end. We refer to those 3' isomers that gain non-templated nucleotides as being tailed. It is mainly uridylation or endogenous. It is well established that microRNA play a critical role in cancer. miR21, for example, function as an oncogenic gene. This plot shows the level of miR21 in multiple cancers. Red bar indicating tumor and green bar represents the corresponding normal tissue. As expected, miR21 level increases during cancer development, which in turn promote cancer progression. Interestingly, the isomer profile of miR21 also changed during tumorigenesis. We found that the level of isomers is different between tumor and normal tissue for several types of cancers, suggesting that those isomers may also have a biological role. Further analysis indicate a major portion of those isomers are tailed isomers. We analyzed hundreds of patient samples from breast cancer, kidney cancer, and uterine cancers. Based on deep sequencing analysis, we calculated the percentage of tailed isomers of all microRNAs. As you can see here, on average, about 20% of microRNA are tailed at the 3' end. We did the same analysis in HEC293 T cells and got a similar result. In this plot, each dot representing one microRNA. As you can see, tailed isomers are highly prevalent regardless of the microRNA abundance. Inspired by those results, we wonder whether tailed isomers are associated with RISC and function in target inhibition. miR27 is known to be uridylated. We therefore use it as a model to investigate the functional impact of uridylation on mature microRNA. This is a northern blot to detect miR27 after immunoprecipitation of Acknowledgements and the GW182, which is a key component of RISC. As you can see here, both miR27 and its isomers were recovered in the pull-down, demonstrating tailed isomers are associated with RISC and capable of target inhibition. However, because 3' modifications do not change the seed sequence, it was believed that the tailed miR27 would regulate the same set of genes as the unmodified miR27. To examine the target specificity of miR27 isomers, we used a dual luciferase reporter assay in which target sequences were inserted in the 3' UTR of the reporter mRNA. When we use seed-matched wild-type target sequences, we observed miR27-mediated repression. When we mutate the target sequence to disrupt the binding at the seed region, repression was abolished as expected. Introducing additional base pairs at the 3' and region resulted in a bold target. And of course, we still observe repression. Disrupting seed binding in this reporter construct resulted in a target site that only paired at the 3' region of miR27. Given the critical role of seed pairing, repression of this reporter should be abolished. However, to our surprise, miR27 was still able to repress this reporter. This observation contradicted the prevailing model of microRNA target interaction. A close examination of the target site reveals that, in addition to the mismatched seed region and 7 base pair at the 3' region of microRNA, there are 2 additional A endosins immediately upstream of the target site. We therefore named this target 7 AA targets. It was possible that the U-tail of miR27 paired to this AA, compensating for the lack of a perfect seed match. To test this, we mutated the AA target to CC target. As you can see here, miR27 was able to repress the 7 AA targets, but not the 7 CC targets, suggesting that uridylate isomers, but not the canonical form of miR27, were responsible for this repression. We wonder whether the pairing between U-tail and the target could be compensated by additional pairing at the 3' region. To do this, we generated a set of reporter forming either 8 base pair or 9 base pair at the 3' region of the microRNA. As you can see here, miR27 was able to repress all the AA targets, but not CC targets. Interestingly, the number of base pair between uridylate miR27 and the 7A target is the same as that of the 9CC target. But we only observe repression on 7A targets, but not 9CC target. This suggests that the potential base pairing between U-tail and AA motif play a unique role in target recognition, which cannot be replaced by simply increasing base pair at the 3' region of microRNA. To directly test whether uridylate isomers are responsible for recognizing those targets, we developed a pull-down assay to isolate isomers physically associated with target mRNA. The idea is to use biotin-labeled probes to pull down the reporter messenger RNA, then analyze associated isomers by northern blot and deep sequencing. Here is the northern blot result. Consistent with the functional data, miR27 can be pulled down from all the AA targets, as you can see here, but not the CC targets. Okay. More interestingly, only the tailed isomers, but not the canonical form of miR27, were enriched in the pull down. We then deep sequenced those small RNAs in those pull downs. We expected that those tailed isomers should be a full-length miR27 with non-templated tails. The first two positions of the tail should be U, which will allow it to base pair with the A in the target sequences. Indeed, deep sequencing results confirmed our expectation. However, when we align all the reads together, we found that enrichment of the U was only observed in the first position of the tail, as you can see here, but not the second position of the tail. In this position, U and A are equally represented. This suggested that a single base pair between mono U tail and the target may be sufficient to stabilize this interaction. I don't have time to show the data, but we performed luciferase reporter assays and find out, yes, a 7A target, which is only 1A in the upstream or target site, can be repressed by miR27. So next, we blocked miR27 uridylation and determined its impact on repression. TUT4 and TUT7 are two enzymes which uridylate both pre and mature microRNAs. We used CRISPR to generating a TUT4 and TUT7 double knockout cells. Deep sequencing confirmed uridylated miR27 was greatly reduced. We then measured the association between miR27 and the 7A targets, use our biotin pull-down approach in either the wild-type cells and the TUT47 knockout cells. As you can see here, the association, what we observed in the wild-type cells, was completely abolished in the double knockout cells. But this association was recovered when either TUT4 or TUT7 was expressed from a plasmid, demonstrating that the TUT47 mediated uridylation on miR27 was required for its association with this type of non-canonical targets. Here is a summary of our results by far. In the canonical microRNA target interaction, seed pairing is critical. However, when miR27 was uridylated at sweeping end, the U tail compared with the target, the A in the target. This enabled the tailed isomer to inhibit messenger RNAs, which normally cannot be repressed by this microRNA due to a lack of a perfect seed match. We named this type of repression Tail-U-Mediated repression, or TUMR for short. Next, we try to identify endogenous targets of this type of repression of miR27. Given our limited understanding of the extent to which mismatches as a seed region could be tolerated, we start our search by requiring only two features. One, a minimal 7 base pair at the sweep region of miR27, and two, an A immediately upstream. This effort resulted in 4,648 target genes. All of them have at least one potential tumor targets within its sweeping UTR. This number is far larger than the number of miR27 canonical targets predicted by target scan, which is about 1,000. We therefore narrowed our prediction to target sites which are relatively conserved. This resulted in about 700 potential microRNA targets. Our bioinformatic analysis predicts there are messenger RNA containing only the seed paired targets, or only the tumor, the non-templated, the Tail-U dependent repression type of targets. and messenger RNA contain both type of targets. To investigate whether those targets are regulated by miR27, we expressed either miR27 or a control microRNA in 23 T cells and compare the level of target mRNAs by RNA-seq. Here's the results. This is the accumulative curve of fold changes between these two treatments. The black line are messenger RNAs without any predicted target size. As predicted, they showed an even distribution of upregulation and downregulation, serving as a baseline. Messenger RNAs bearing seed-paired canonical target sites are indicated by this red line. They shifted to the left, indicating that as a group, they were downregulated by miR27. The blue line are those with tumor target sites. They were also downregulated by miR27. Interestingly, those messenger RNAs containing both types of target sites, indicated by this green line, are the most repressed by miR27, suggesting there is an additive effect between the canonical and the non-canonical form of microRNA repression. We repeated the same experiments in TUT4 and TU7 double knockout cells. The repression on messenger RNA containing only the tumor target sites were completely abolished, as the blue line representing messenger RNA with tumor target sites now superimposed with the black baseline. In addition, the green line showing those with both types of target sites overlaps with the red line, which is the ones only have the canonical sites, indicating the additive effect was also abolished. These results confirm that on a transcriptome scale, Tail-U-mediated repression is dependent on the activity of tailing enzyme TUT4 and TUT7. Next, we ask whether other microRNAs besides miR27 mediate tail-dependent repression. To test this, we compare the isomer profile between wild-type cells and the corresponding TUT47 double knockout cells. The result is plotted here. The y-axis is a change of uridylation, and the x-axis is the isomer abundance. We found that the uridylation of miR148B was dramatically reduced upon TUT47 depletion, suggesting that miR148B has the potential to regulate a set of endogenous targets via Tail-U-mediated repression. Indeed, when miR148B was blocked by an antagomer, we found that messenger RNAs harboring the predicted tumor target sites, which shown by this blue line, were de-repressed. And once again, we observed the additive effect between canonical and tumor target sites, shown by the green line. In parallel, we used probes against poly-A tail to pull down all cellular messenger RNAs and analyze the isomers presented in the pull down by deep sequencing. The idea is that those target-associated functional isomers will be enriched. The result is plotted here, each dot representing one microRNA. The x-axis indicating the enrichment in the abundance comparing pull down with input, while the y-axis measures the enrichment of uridylation. As you can see, many uridylated microRNAs were enriched in the pull down. As expected, miR148B was here. We also detected other uridylated microRNAs, such as miR34A. This result indicates that those microRNAs may also regulate a set of endogenous targets via Tail-U-mediated repression. So why does a single base pair between the U-tail and the target could make such a big differences? Here is our proposed model. Based on crystal structure of RISC complex with target, we know that there is a seed chamber, which explains why seed pairing is essential for this interaction. Recent structure study revealed there actually is another additional chamber called supplementary chamber, where the pairing between the 3' end of micRNA and the target can occur. We hypothesize that the pairing between the tail of micRNA and the substrate could facilitate the formation of this supplementary chamber. In this way, pairing of the tail would contribute more than just additional binding energy. It would also shift the mode of target RISC interaction to involve this supplementary chamber. Of course, future structure and biochemical study will be required to test this hypothesis. In summary, our finding revealed a new way in which micRNA selects their targets. Canonical micRNA regulate a set of genes through seed pairing. But after tailed by TUT4 and TU7, those uridylated isomers would be able to regulate a new additional set of genes through this type of non-canonical tail-dependent repression. Our results also implies that many micRNAs have additional targets that are not predicted by commonly used softwares. Besides 3' isomers, we also study the biogenesis and function of 5' isomers, which I don't have time to talk about today. Given that 5' isomers have a different 5' end, it is easy to understand that those isomers modulate RISC target interaction by shifting the seed sequence. This change in seed sequence will allow 5' isomers to regulate a distinct set of target gene and therefore have unique biological function. Hopefully, I convinced you today that micRNA can diversify its function via sequence modifications at both the 5' and 3' ends. It is well established micRNA play a critical role in cancer. We also know a lot how micRNA are regulated, their abundance is regulated in cancer. We propose that sequence modification, such as tailing, also regulate micRNA function in cancer. There could be oncogenic isomers or tumor suppressor isomers. Finding those isomers will explain why specific isomer profiles are correlated with a different stage of tumor progression. To this end, we analyzed small RNA-seq data from TCGA and target projects. Those are very comprehensive data sets, which includes about 100 billion reads collected from over 10,000 patients in 36 types of cancers. By those analyses, we established a Tumor IsomiR Encyclopedia, which is hosted by NCI. Here is a screenshot of the website. I encourage you to give it a try and check out the isomers of your favorite microRNA. This work was done by two talented fellows, Dr. Ah-Tsung Yang and Dr. Xavier Barfield-de-Ross. I will stop here and happy to answer any questions.
So thank you, Shua. That was a wonderful talk. So we already have a question in the chat from Mills Walter. Mm-hmm. Does suppression of the target by a 3' U modified microRNA still require recruitment of the CCR4 NOT complex and target degradation starting from the 3' end of the mRNA?
So we do not have evidence, but I would assume, yes, they will still require that. Because again, I think those factors function downstream of RISC target association. So yes, so they will require those.
We have a question from Tang. Does your model predict tailing occurs prior to microRNA target interaction? Or could tailing occur while microRNAs are associated with their targets?
Actually, this is a great question. Actually, this is the answer we try to, this is the question we try to answer. So we think both are possible, right? But given actually the uridylated isomer for each particular microRNA is relatively low. So if those uridylation can happen locally after they recognize the suitable targets, actually that will indicating those uridylated isomer have a bigger role. okay, in gene regulation. So currently we are testing, we are doing experiments to answer the question, but right now I do not have the answer. So both are possible.
We have a question from Frank Slack. Were you able to validate any novel miR27 or miR148 targets in cells?
Yes, actually that's what we are currently doing. And we try to go into looking after those which we potentially have a biological role. So for example, the one potentially with 34A or from other microRNAs and in the content of the cancer cells. But right now I don't have time to show those, but hopefully next time when we present our work, we can show those results.
So here's another question. When a tail is added, shouldn't that reduce the number of targets and be a subset?
No, we don't think. So we think actually the tailed one was still able to go through the canonical repression, right? Because they still have the seed pairing. So actually instead of loss their ability to repress canonical targets, they only gain ability to of this new set of non-canonical targets.
So we have another question. What percentage of microRNAs have 3' U modification? Okay. And does the overall modification level correlate with the expression of TUT4/7?
I think this is another great question. So first of all, I think that depends. Each different microRNA have a different percentage of uridylation, and also that depends on the content of the cell. In different stage, actually that's different. So right now, I think the two major enzymes, as we showed in this study, would be TUT4 and TUT7, right? Those two enzymes are adding U. But actually, we have additional evidence indicating, actually TUT1 would also potentially adding U at the end. So yes, we already checked. So I think I may have the data here. So actually, we're even looking for like different type of cancer at the level of TUT4, TUT7, and TUT2 are different. And so they definitely are different. So actually, that's corresponding very well with the overall uridylation of those isomiRs. So we think that could potentially play a role in this type of regulation.
Does this mechanism fully depend on tailing, or can 3' end U-rich microRNAs work similarly?
So I really thinking, okay, so I think the question is similar to the question asking before, right? So when the U-tailing happens, right? So do you need the TUT4/7 to be there to stabilize the complex, right? Or you can already born with the U at the end and you can still able to go through this pathway. So I think the short answer is we don't know. That's the things we try to understand. We're doing lots of biochemical approach and try to do some structure, try to gain some structural insight. So by then we can answer this question.
And one more question. Do you observe any cases of differential isomiR expression between normal and tumor cells?
Yes, actually that's answered yes. So globally, definitely we see lots of differences, and in individual cases, we see differences. So, but right now, what we're doing here is, we can say on global level, the isomer profile is definitely different, right, in speaking of uridylation. But how the difference is actually also vary between each microRNA. So certain microRNA have big difference in speaking of uridylation, and some microRNA have not much of differences. So that's something we're testing right now. We're thinking those have bigger differences in uridylation would potentially have a role to play like during tumorigenesis.
Okay, so thank you, Shua, for a great talk. If anyone has any more questions, they could put them in the chat and Shua can answer them later.
We're not happy to.
So our next speaker is Dr. Stavroula Milley. She's an investigator in the Laboratory of Molecular and Cellular Biology at the National Cancer Institute and National Institutes of Health in Bethesda. So Voula was an undergraduate at the National Kapodistrian University of Athens in Greece, a graduate student at the Mount Sinai School of Medicine in New York, and a postdoctoral fellow with Joan Stites at Yale and Ian Macara at the University of Virginia. And she has been at the NIH since 2012. So Voula.
Great, thank you very much, Andy, and thank you very much for giving me the time to tell you some of our work. So my group is interested broadly in localized RNAs, And today I will be telling you a study about how RNA localization can impact on protein function. So generally, localized RNAs are RNAs that are targeted to particular regions in cells. And they have been quite extensively studied in model organisms, such as in budding yeast, where the ASH1 mRNA is a classic example. In Drosophila oocytes, where axis specification is determined largely by localization of RNA such as bicoid or OSCAR, and in mammalian neurons, where a large number of transcripts are targeted in axons or dendrites and are important for axon outgrowth and synaptic plasticity. Now, in these types of systems, usually localization of an RNA leads to a corresponding accumulation of the encoded protein. So for example, translation of the localized OSCAR RNA leads to a corresponding increase in the OSCAR protein at the anterior of the oocyte. And translation of dendritic RNAs upon synaptic stimulation is thought to lead to accumulation of proteins there that are underlying synaptic plasticity. And indeed, more global correlation screens have shown that there is a significant correlation between RNA and protein enrichment in these systems. Now, we are looking at localized RNAs in a different context, which is in somatic non-neuronal mammalian cells. These cells are smaller, less morphologically polarized, and much more dynamic than neurons. And they include cells like fibroblasts or cancer cells. In these cells, RNAs can be targeted in a variety of locations. We are mostly interested in RNAs that are targeted to these protrusive regions as the cells are migrating. One such RNA is the beta-actin RNA, which is studied quite a bit. However, we had also shown that there is a large number of other RNAs that are targeted in these protrusive regions, such as this green RNA shown here in these in situ hybridization images. And these are targeted prominently at these protrusions, and this distribution is in contrast to that exhibited by several other RNAs, which are much more diffuse in the cytoplasm. We had identified a large number of such protrusion-localized RNAs using these protrusion cell body fractionation scheme, and several other studies since then have shown the same thing. The interesting thing that we noticed, and that other people have reported also, is that in these cases, there appears to be little correlation between where the RNA is and where the protein ends up. So you can target an RNA at the periphery without necessarily accumulating the protein there. So what I will be telling you about today is a study that tries to understand why that is so. Why would you need to actively target an RNA to a location if not to concentrate the protein there? And as a case study, I will use the RAP13 RNA. So all these genome-wide screens identifying protrusion-enriched RNAs have varying degrees of overlap. But there are several RNAs, some of them are shown here in a partial list, that are consistently and prominently enriched in protrusions of various types. And at the top of the list is RAP13, and for this and for other reasons, we're focusing some of our attention on it, and this will be the focus for today. So yeah, what is RAP13? So RAP13 belongs to the RAP family of GTPases. As such, it cycles between a GTP-bound active and a GDP-bound inactive state. As other RAP GTPases, it is involved in vesicle trafficking and endosome recycling, and it has been reported to have various cargoes or effectors, such as integrins and the cytoskeletal protein filamin. These type of effectors control adhesion and cytoskeletal dynamics, which are processes involved in cell movement. And consistent with that, RAP13 has been shown to be important itself for cell migration. So for example, what I show you here is that, like other people have reported, if we knock down RAP13 using siRNAs, we see a reduction in cell migration speed. We can rescue this effect when we re-express the full-length wild-type RAP13 RNA, but not when we re-express the same RNA that carries a frame shift mutation that prevents production of the protein. So this is just to state from the beginning that the RAP13 RNA on its own is not sufficient to support cell migration in some potentially non-coding or translation-independent role, but you actually need to produce the RAP13 protein for efficient migration. So keep that in mind. So the RAP13 RNA, though, shows this interesting characteristic I mentioned, that it is preferentially enriched in these protrusive regions. This is not a peculiarity of cells grown on these two-dimensional glass surfaces. We have looked at multicellular cancer spheroids that we embed in three-dimensional matrigel. We can induce them to extend to invade into the surrounding matrix. And what we notice again is that there is this striking accumulation of the RAP13 RNA right at the front of the invasive leader cell. We have also looked at tumors in vivo. This is a tumor grown in the tongue of a mouse. This is an invasive tumor mass, and the green signal here highlights the tumor cells into the surrounding normal mouse tissue. And when we focus on these potentially invading tumor areas, what we see again is that the RAP13 RNA, shown here in red, seems to accumulate again at the front of these potentially invasive cells. So localization of RAP13 can be seen both in vitro and in vivo, and it seems to happen at the front of cells that are migrating or invading. Now, we have tried over the years to understand what are the underlying mechanisms that target RAP13 and other protrusion RNAs at this periphery. I will not go through any of these today. But what is relevant for today is that we have also looked at how and when the RAP13 RNA is translated using either biochemical or imaging tools, such as these SANTAG reporters shown here, which allow us to visualize single RNA molecules and to image at the same time if they're actually being translated and produce nascent chains. So I won't have time to go through the details. This has been published recently, but the point that is relevant for today is that RNAs that contain the RAP13 regulatory sequences, when they're found at these peripheral extending regions, they're actively being translated into protein. So the question that we want to address today is, why do you need to target the RNA there and translate it there? And I told you that in many cases, there is a discrepancy between RNAs that are localized in a particular location and where the protein is distributed. And this is the case for RAP13. So even though the RNA is peripheral, and as I just told you, we think it is translated there, the protein ends up being around the nucleus. And these are just controls for the specificity of the immunofluorescence signal here. Now, we see the same result, the same situation when we look at isolated protrusions in cell bodies. Again, the RAP13 RNA is enriched in these protrusions, whereas the RAP13 protein is not. So to understand why you would need to target the RNA in the periphery, we needed to find a way to prevent the RNA from localizing in the periphery without, though, altering the RNA levels or the amount of protein that is produced from it. And we knew that the 3' untranslated region of the RAP13 RNA is important for localization. So we tried to understand a little bit more what are the important factors in it. And we noticed this GA-rich region. So this is a plot of the GA content along the 3' UTR of either the mouse or the human RAP13 RNAs. And there is this extended region at the beginning of both of these UTRs that are very high in GNA nucleotides. And there are these motifs that can be recurring and can be noticed in these GA-rich regions. These are these red boxes here. And indeed, if we delete some of these motifs, we see that we reduce localization of the RNA at the periphery. Now, apart from this deletion analysis, and because we were mostly interested to mislocalize the endogenous RNA, we have been mostly using this antisense oligo-based approach. Specifically, we're using morpholino oligonucleotides that we target against specific regions in 3' UTR, with the idea that if you target a functionally important region, either by disrupting secondary structure or by preventing binding of a protein factor, you might interfere with localization. So here we're tiling the whole 3' UTR of the human RAP13 RNA with different oligos. We deliver individually these oligos, and then we assess RNA distributions from this type of in situ hybridization images. And we quantify RNA distributions measuring what we call a peripheral distribution index, or PDI. The higher the index, the more peripheral the RNA, and an index of 1 indicates an RNA that is completely diffuse. So what we see is that if we deliver oligos that are targeting the GA-rich region, this leads to a reduction in the peripheral accumulation of the RAP13 RNA. We can deliver two different oligos at the same time and get an additive effect, which basically converts the peripheral localization of the RNA to a more perinuclear one. This effect is specific, since if we look at the net1 RNA, another peripheral RNA within the same cells, its distribution at the periphery is not affected. But most importantly, if we look at the amount of protein that is produced from this mislocalized RNA, it is not affected. So in here, we are delivering one oligo against the GA-rich region, or the combination of oligos that have an even more pronounced effect on localization. And in all cases, the amount of protein that is produced from these RNAs is unaffected. So we looked also more broadly at the specificity of this effect. Here we are isolating protrusions and measuring enrichment of a panel of different RNAs in these isolated protrusions, either in control cells or in cells where we have delivered these antisense oligos against RAP13. What we see is that only the RAP13 RNA, the RNA that is being targeted, is affected in its enrichment in protrusions, whereas all the other RNAs remain unaffected. We have also looked at overall RNA levels, and we see no difference when we deliver these antisense oligos. So our conclusion is that this antisense oligo-based approach allows us to convert the RAP13 RNA from a peripheral to a more perinuclear distribution, and we can do that without altering RNA levels or the amount of protein that is being produced. So now this allows us to address what are the functional consequences specifically of altering the location of the RNA. And since RAP13, the protein is involved in cell migration, we tested if there are any consequences in the ability of cells to move. We looked at a variety of different assays. Here we're looking at the dynamics of the cell edge. This is a transwell migration assay where we're measuring how efficiently the cell moves towards a chemoattractant. This is a random migration. We're measuring the speed with which cells are moving. And here we're inducing the cells to invade through a three-dimensional matrigel. And in all cases, when we deliver oligos against RAP13 and prevent the peripheral localization, this leads to a reduction in the ability of cells to move or invade. So this is good. This suggests that there is a functional requirement for targeting the RNA to the periphery. However, what I think is even more striking is the extent of this defect. So if you remember, I showed you in the beginning that when we knock down RAP13 with siRNAs to almost undetectable levels, we reduce the ability of cells to migrate, we reduce their migration speed, as well as the dynamic of the cell edge here. Now, if we just deliver antisense oligos against RAP13, which simply mislocalize the RNA, we get a defect to the same extent. And the remarkable thing is that this happens even though the amount of protein that is being produced from this mislocalized RNA is virtually the same as the one under normal conditions. So this suggests that the protein produced from a mislocalized RNA is virtually non-functional with regards at least to cell migration, and it is as if the protein was completely absent. So then the question then becomes, what is different between the protein produced from a mislocalized RNA, and what are the properties of the protein that are affected based on the sites of its synthesis? So what we'll be showing you in the next few slides is we characterized either the protein produced from a mislocalized or from a correctly localized RNA. We do that either for the endogenous protein, as seen here, upon delivery of antisense oligos, and as an alternative approach, and to avoid potential off-target effects of these antisense oligos, we have also been characterizing exogenous protein. In this case, we are expressing GFP-tagged RAP13 that is expressed from a construct that carries the full-length wild-type 3' UTR of RAP13. This is localized in the periphery, as quantified here. And we compare this protein with a protein produced from a similar construct that only has a deletion in the 3' UTR of the region that is targeted by the antisense oligos. So this construct, as expected, is perinuclear, as quantified here. The RNA of the two constructs, the RNA levels are the same, and the amount of GFP RAP13 protein produced from two constructs is the same. as quantified here by flow cytometry or by Western blot analysis. So before I go into our characterization of these two different types of proteins produced from a localized or mislocalized transcripts, I need to give you a bit of an overview of RAB-GTPase biology. So newly synthesized RAB proteins are thought to associate with proteins called RAB escrow proteins or RAPs. These bring the newly synthesized RAB protein to the geranyl transferase complex that modifies them with lipids and targets them to the membrane. There, the protein cycles between an inactive or an active state, which is loaded with GTP. And this cycle is catalyzed by the action of activators called guanine nucleotide exchange factors or GEFs, or the protein is inactivated through the actions of GTPase activating proteins or GAPs. In its active state, the RAB protein binds to effectors, which mediate the functional outcomes, and the protein can also be extracted from the membrane and be kept soluble into the cytosol through the action of proteins called RAB-GDIs. Now, knowing all this, we try to characterize the RAB13 protein produced from normally localized or mislocalized RNA with regards to its steady state distribution, its association with membranes, interactions with regulators, or its GTPase activation status, its loading with GTP. And I will go pretty quickly through most of this. With regards to steady state distribution, we looked at endogenous or exogenous RAB13 by immunofluorescence. Basically, we see no difference. We looked at membrane association by fractionating into a particular or soluble fraction. Again, we see no difference. We looked through immunoprecipitation for association with RAB-GDI or RAP. Again, the two proteins are the same. But we see a difference when we looked at the level of activation of the protein or its the amount of loading with GTP. The way we detect that is by using a pull-down assay with the domain of one of its effectors that binds specifically to GTP-loaded active RAB13. And what we see is that when we look at the endogenous protein, when we deliver the antisense oligos and mislocalize the RNA, the level of activity of the protein is reduced. And similarly, when we look at exogenous RAB13, when it is translated from a mislocalized RNA, it is less active. So this suggests that you need peripheral localization of the RNA to get full activation of the protein. Now, I told you here that to activate the RAB-GTPase, you need the action of GEFs. So to understand the mechanism a bit better, we try to identify what proteins might function as GEFs for RAB13 in the system. And to do that, we use the mass spectrometry approach. We try to identify proteins that bind specifically to this mutant form of RAB13, this T22-2N mutation. This mimics a nucleotide-free intermediate that has high affinity for GEFs. And indeed, through this approach, we found this protein that binds tightly to this mutant, which is called RAB interacting factor or RAB-IF. This is a protein that has been previously reported to function as a GEF. So we validated these findings, and indeed we see by immunoprecipitation that RAB-IF binds to the wild-type RAB13 protein. It binds with higher affinity to this nucleotide-free mutant, as would be expected from a GEF, which is quantified here. Also, if we knock out RAB-IF expression using CRISPR and two different guide RNAs and look at RAB13 activity, we see a significant reduction. So this suggests that indeed RAB-IF can function as a GEF for RAB13 in the system. The question, though, that was relevant for us was whether RAB-IF is part of this location-dependent activation mechanism. And indeed, we see that RAB-IF binds less to RAB13 when RAB13 is expressed from a mislocalized RNA. So this would indicate that you need peripheral translation of RAB13 to form more efficiently the complex between RAB13 and its activator RAB-IF. Now, one potential way that this could happen would be if RAB-IF was more enriched in the periphery, and therefore if RAB13 is translated there, then the two proteins can interact more efficiently. However, this doesn't seem to be the case. As I showed you for RAB13, also RAB-IF is more perinuclearly enriched with small amount of the protein reaching the periphery. So this couldn't explain why you need to translate RAB13 in the periphery for the two proteins to interact. So to understand this better, we try to see where exactly in the cell are the two proteins actually interacting. And for this, we use the proximity ligation amplification, or PLA assay. And the red PLA dots here show where in the cell the two proteins are interacting. The signal is largely specific because it is reduced when we knock down either one of the two interacting partners. Now, we did this experiment looking at both interaction between RAB-IF and endogenous RAB13, or interaction between RAB-IF and the exogenous GFP-RAB13, and we basically see the same thing, which is that the two RAB-IF interacts with RAB13 both at the periphery, but also in more internal locations in the cell body. But interestingly, when we mislocalize the RAB13 RNA, we disrupt specifically the peripheral interaction as quantified here, whereas the more intranuclear, the more internal cell body interaction remains unaffected. So this indicates that this peripheral interaction between these two factors is directed by where the location of the RNA is. And one way that we thought this could happen would be if the two proteins are interacting during translation. So with RAB-IF interacting with the nascent RAB13, and indeed, based on the structure of other RAB GTPases, the domain within RAB13 that is bound by RAB-IF is at the N-terminus, suggesting that this interaction could actually happen during translation. So to see if there is really a co-translational interaction, we immunoprecipitated RAB-IF and tested to see whether we pull down the RAB13 RNA. And indeed, we see a specific interaction between the two. And if this model is correct, then the expectation would be that if you disrupt ribosomes with puromycin, this interaction should be abolished. And indeed, this is exactly what we see. So to put everything together, we think that you need to translate, to localize the RAB13 RNA in the periphery, not simply to concentrate the RAB13 protein there, but to allow an interaction between the nascent RAB13 with its activator RAB-IF within this particular local environment. And this peripheral interaction targets the RAB13 GTPase to support cell migration. And preventing this peripheral interaction, co-translational interaction, disrupts cell migration to the same extent as if the protein was completely absent. Now, when we disrupt this peripheral mechanism, though, we see a significant amount of active RAB13 still present. So we think that there are additional GEFs, activators, that probably target RAB13 to support processes other than cell migration, probably by coupling into different effectors. And I also told you that there is a perinuclear RAB13 RAB-IF pool that is not again affected by this peripheral mechanism. And we think this might have to do with another reported role of RAB-IF as a chaperone. However, apart from these details that have more to do with RAB GTPase biology, we think that the more general take-home messages of this work is that the location of the synthesis of a protein within the cytoplasm can be important. And simply by altering the location of an RNA, you can affect the regulation or the activity of the encoded protein. Given that there is a large number of transcripts that show localization to particular sites and a discrepancy with where the protein actually ends up, we think that this mechanism might be more broadly applicable to additional proteins apart from RAB13. So I will leave you with that. And I'll just like to thank the people who were involved in this work. So this work was mostly done by Costas in the lab with help from Michael, T, and Alex, who is now also looking at another interesting mechanism we think that connects localization with protein function. We're supported by core facilities within NCI. Michelle Hastings was very helpful for our initial design and understanding of the use of antisense oligos. Many thanks to the NCI and the Center for Cancer Research for generous support, and to the RNA Biology Initiative for making it a very nice place to work in. And thank you all out there for taking the time. Thanks.
So thank you, Vula. So we have a question from Susan Gottesman. Do you think the rate of protein translation and thus folding changes with changed mRNA localization? this could affect the ability to interact with the RAB1F? Or is there another explanation for why RAB1F prefers to interact at the periphery?
So, if I understand, so I don't think there is a change in the rate of translation because we don't see any change in the overall levels if we alter the localization, unless it's a very slight one. So we don't necessarily think that there is a change in the rate of translation. And there is still interaction that occurs in the periphery. So it doesn't, the interaction doesn't only occur at the peripheral locations. We think that there is probably some other local event that happens that is part of this mechanism, which could involve either local modifications or the presence of local effectors. So even though the TTPase might be active in the periphery, I guess its eventual function will be affected by what type of effectors it is coupled to. So we think that there are additional components of this mechanism, but we don't have any evidence that the rate of translation would be different in different locations.
So we have a question from Zhangyu Liu. Is there any data about what proteins or RNAs the GA-rich region can interact with? Do other mRNAs have a similar sequence in the 3' UTR?
So we're looking at what proteins are binding. So this particular GA-rich motif is found in other protrusion-enriched RNA. So there is RAP13 belongs to this group of RNAs that we think are co-regulated, and they depend on some of the same factors, such as the APC tumor suppressor protein and the tyrosynated microtubules. All of these this group of co-regulated RNAs appear to be enriched for these GA-rich motifs. And we have been looking at what protein factors associate with them or what kinesins are involved. And yeah, we're using the standard kind of pull-down mass spec analysis to try to find proteins. But yeah, all of this work is in progress right now.
We have a question from Christina Fitzsimmons. Do you observe any changes in post-translational modifications that depend on the localization?
We haven't yet. We've looked. So as I mentioned, yeah, we think that maybe there is probably some component of probably different modifications or something else that could be overlaid on this mechanism. We have looked, but we haven't found anything that seems to be relevant yet. But yeah, it's not an exhaustive analysis yet. So we're still looking towards that.
We have a question from Peter Johnson. Do you think the antisense oligo is disrupting a protein RNA interaction or a 3' UTR structure?
We don't know yet. It could be either, I guess. So that's why we're trying to identify some of the proteins that bind. So we have some candidates, and we're not trying to see, if the oligos would actually displace those. But I guess it could be, yeah, either one. We don't know yet.
So Rich Mariah would like to know if you can develop a RAB1F construct with a localization signal that can rescue the GTPase deficiency of the localized mRNA?
Yeah, that could be interesting. Yeah, we haven't thought about that. I guess it would depend on, because with our model, the co-translational interaction probably happens on the nascent RAP13. It's not clear if the RAB-IF is fully, if it's the fully mature protein that interacts with it, or if it happened, if both of them need to be translated at the same location. But that could be an interesting thing to try out.
And finally, what's the role of mRNA decay mechanisms on the localization of RAB13 mRNA? Are the localized mRNAs protected from degradation?
We don't have any evidence for that. So what we know is that if we prevent the localization, we don't change the RNA levels. So it doesn't seem that they are degraded if they don't go there. I mean, I don't know what, so these RNAs and a lot of these protrusion RNAs that belong to this co-regulated group are all very low abundant. So there are only 5 or 10 copies per cell for some of them. So it's not clear what exactly keeps their copies so low. But we haven't seen that the degradation is coupled to the localization itself. But it could be independent of that.
So now we're ending exactly at 5 o'clock, perfect timing. And this will be posted on YouTube. And I want to thank both speakers for fantastic talks. And I also want to thank all of the audience for participating. And as always, the next talk in this series will take place in 2 weeks. So see you there.